Yeah, the question is, in which genus glycine is absent in their cell wall peptide bridges? So that is the question. They are giving the options: Nocardia, Streptomyces, Frankia, Monospora. So all these options are related to Nocardia, Streptomyces, and Frankia, Monospora. These are all related to Actinomycetalis. Okay, Actinomycetalis, and they belong to Actinomycetes class. Okay, Nocardia is a saprophyte. Actually, these are all the gram-positive bacteria, but they have a specific feature like mycelium. So they are called as actinomycetes. Okay, and they belong to the order Actinomycetalis. These are all bacteria, but they form the mycelium. That is the common feature. Nocardia, it is a saprophyte. Streptomyces, it is a saprophyte. Frankia, it is a symbiont, okay, in legume. So it forms symbiotic relationship with the non-legume plants like Casuarina, okay, and it is useful for the nitrogen fixation. This Frankia is useful for the nitrogen fixation, so it is very important organ. And also micromonospora. So they just gave the monospora, but it is also called as micromonospora. It is also a pathogen. It acts either as pathogen or saprophyte. So this is all about the options they gave. So in which genus glycine is absent in their cell wall peptide bridge? So actually the cell wall of the bacteria contains the peptidoglycan. Okay, peptidoglycan. And also they have peptide bridges in the cell wall. Usually the cell wall is very important feature to distinguish the plants and the bacteria. Plants also have the cell wall, but they are made up of cellulose, hemicellulose, pectin, lignin, all these are the material. But the cell wall of the bacteria contains the peptidoglycan. Peptidoglycan is also, they contain the components like NAG, N-acetyl glucosamine, and NAM, that is N-acetyl muramic acid. So these two components are cross-linked. So you should read the cell wall structure, okay? And also you have to read the cell wall structure of the gram-positive bacteria and gram-negative bacteria. These are very important for the competitive exams. And the cell wall contains the peptide bridges, and in the peptide bridges, glycine is absent. In which of the organisms, in which of the actinomycetes, or because all these belong to actinomycetes only. Okay, so Nocardia is the answer for this, because Nocardia lacks glycine in the peptide bridges of the cell wall. So the right answer is Nocardia. So once you have to go through the different kinds of the actinomycetes, okay, and what are the features of the actinomycetes in your first semester botany. So next question is, the wall-less prokaryote confined to the phloem of plants is known as, okay, microbial diversity. In the first unit, you read this one. What is the wall-less? That means until now we have discussed about the cell wall of the bacteria, which contains a peptidoglycan. Okay, but in case of the prokaryotes, bacteria also prokaryote, but these are also the prokaryotes. They contain nuclear material, protoplasm, everything, but and they have the cell membrane also, but they don't have cell wall. So what are the organisms? Actinomycetes? No, actinomycetes, They have the cell wall. Just now we discussed. They also have the cell wall made up of peptidoglycan. And bacillus is also bacteria, eubacteria. It contains the peptidoglycan cell wall. And virus, it is not at all related to these microbes. They don't have virus, they don't have plasma membrane, they don't have cell wall. Okay, and it is not belong to prokaryote. The question is about the prokaryote. So virus is entirely different organ. Okay, so it is also wrong option. So the right option here is given as mycoplasma. So mycoplasma, these are also called as jokers of the plant kingdom. Okay, they lack the cell wall. And one more feature is mycoplasma. These are also initially called as PPLO, pleuropneumonia like organisms, PPLO. Okay, now caught. He described that. And PPLO, they are also called as, and they cause several diseases, pneumonia. The diseases which caused by mycoplasma are mostly related to the lungs. Pneumonia, they may cause pneumonia. So pleuropneumonia like organisms they are called. So now the question is about the wall-less prokaryotes confined to the phloem of plants. But one more thing you have to remember, there will be one more question. Actually, mycoplasma, in our books, it is given as mycoplasma, but it should be like phytoplasma. Sometimes they may ask, instead of mycoplasma, they may ask phytoplasma, because mycoplasma and phytoplasma both are related with respect to cell wall. Both of mycoplasma and phytoplasma both don't have the cell wall. But mycoplasma is mostly related to the organism which causes the disease in the animals and human beings. But whereas phytoplasma causes disease in the plants. Okay, but in our books only they gave about the mycoplasma. But sometimes they may ask about the phytoplasma also, which particularly causes the disease in the plants. And it grows in the phloem. Since if they give mycoplasma and phytoplasma, if they give the both the options, then you should choose phytoplasma instead of the mycoplasma, because we have to choose according to the nearest answer, whichever the nearest answer. Since they didn't give the phytoplasma, we have to choose the mycoplasma. If they gave both mycoplasma and phytoplasma, we should choose phytoplasma only. Both don't have the cell wall, but phytoplasma is related to the plant diseases like the little leaf of brinjal. And mycoplasma is mostly related to the animal diseases. So next, and one more thing you should remember, they may ask one more important question, that is the, in which of the organisms penicillin does not work? Because penicillin, it is an antibiotic. It was discovered by Alexander Fleming. It was extracted from Penicillium notatum. Penicillium notatum is the fungus from which he discovered the penicillin. That compound was isolated from Penicillium notatum. That penicillin is the antibiotic. It acts particularly, specifically on the cell wall. Okay, that you should remember. So since these mycoplasma or phytoplasma don't have the cell wall, they can't harm these organisms. Okay, penicillin cannot act on mycoplasma and penicillin cannot act on phytoplasma because they don't have cell wall. That you should remember. And one more one, during which geological time cyanobacteria originated. Okay, so you might have read about the geological time scale. They have given the options Cambrian, Devonian, Silurian, and Archean. So just we'll once go through about this geological time scale. Here you see, if you see the most primitive time period: Hadean, Archean, Proterozoic, and after that, you see this Ordovician, Devonian, Permian, Triassic. All these periods are given in the time scale. So here the right answer is, see, Proterozoic. In the Proterozoic era, bacteria, algae, jellyfish, all these were originated, and before that only cyanobacteria is more primitive compared to the bacteria, algae, and jellyfish. Okay, so it belongs to the period Archean period, that you have to remember. So that is the right answer for this question. During which geological time cyanobacteria originated? Right answer is Archean period. Okay, next. In which type of fossils plant anatomical details are more or less preserved? So once again we are discussing about the fossils. Okay, there are different types of the fossils like compressions, incrustation, impressions, and petrifaction or petrification. Anything. So here, compressions, um, instructation, and impressions, all these give the external morphological features. If you want to study the anatomical features, you have to go for petrifactions or petrifications. So already once we discussed about this one, you see these are all different kinds of the fossils: compressions, impressions, and castings, and petrification. So this is the petrification. Here we see the enlarged one. So here in the petrification, what happens? The plant tissue. In the plant tissue, the organic material is replaced by the silica or mineral matter in the soil. So here the anatomical features are kept intact because since this organic matter is replaced by the inorganic mineral matter or silica, this this original form of the plant is kept intact for millions of years, because there is no organic matter here, because so there is no choice for the decaying of the organic matter here, because the organic matter, if there is any organic matter only, it decays by the bacterial or any action. But since there is no organic matter here, and it is replaced by the mineral matter, so we can see all the anatomical features here. So that is the importance of the petrifactions or petrifications. Okay, and that is the right answer. Okay, in which type of fossils plant anatomical details are more or less preserved? So the right answer or most suitable answer is petrifaction. And if you see the compressions, impressions, they give the external morphological features. You see, as we discussed, you see here, this is the impression, just like a thumb impression. If you press something, if you press your hands on top of the soil, you get the impression. Okay, and compression, it is a compressed material. The external soil, what happens? The any living organism or any dead organism which is in the inner layers of the soil, it is compressed by the soil and it forms a structure and it gives the external appearance. We can see the external appearance of that organism from the outside, not from the inside. And also castings also it gives the external features only, not the anatomical or internal features. The only way to study the anatomical features is by studying the petrifaction. Okay, that is the right answer. Next. So next one is rostellum is a special structure present in the family. Now we come to different types of the families where we see some specific structures. Rostellum, in which family we see this structure? In the flowers of the Orchidaceae. So they gave the options like Euphorbiaceae, Caparidaceae, Astraceae. But in case of the Orchidaceae, we see a specific structure, floral structure. And there are specific structures here, like rostellum, where it is present. Rostellum is a structure which separates the pollinia. In the in the Orchidaceae family, one more important, it is very important with respect to competitive exams, Orchidaceae family, because it has so many special features, like the formation of the pollinium. What is pollinium? Where we find in only two families, in Asclepiadaceae and in Orchidaceae, we find the pollinia, plural form. Pollinium and pollinia is the plural. Here what happens? Pollen grains, they form a segregated structures, ball-like structures. Okay, they stick together to form a structure. Several pollen grains stick together to form a structure called as pollinium. So that we observe in case of the Orchidaceae and also in case of the Asclepiadaceae. And also specifically in case of the Orchidaceae family, we find this rostellum structure. So this rostellum, what it where it is present? It present between the pollinium and the stigma. It is the stigma. Outside structure is called stigma. In between the stigma and pollinium, this rostellum is present. It makes sure that there will be no self-pollination. The main function of the rostellum is to form a barrier between the stigma and pollinium, and it prevents the self-pollination in the Orchidaceae family. And one more important feature of the Orchidaceae family is that there is a special attraction that is called labellum. It is a lip-like structure, and due to the presence of the labellum, it loses its symmetry. So in the floral diagram, in the floral formula, we have to mention that whether it is symmetric or zygomorphic or asymmetric. Due to because it is an odd petal labellum, it is formed actually. It is present in the posterior side, but due to the torsion, దీన్ని మెలిక అంటారు, torsion or inversion, the upper labellum, it takes 180 degrees turn and it comes down. Okay, the entire floral structure, it is inverted. That is called as torsion. It takes 180 degrees. The upper structure comes to in the downward direction. That structure is called labellum. It is useful for the attraction, to attract the pollinators for the pollination. That is the function of the labellum. So you have to remember, one is rostellum, another is the pollinium, and labellum. What is torsion or inversion? So all these terms you have to remember, you have to study. And next, so here the question is, rostellum is a special structure present in the family Orchidaceae. And also one more structure I want to tell, that is a gynostemium. Okay, gynostegium, gynosteria. These are the two different forms. Again, gynostegium is related to Asclepiadaceae and gynostegium is related to Orchidaceae family again. So you have to study all these topics. Next, what is the basic unit of inflorescence in OAC? Now they asked one more question from the families. OAC. And in POAC, simple raceme, cyme, head inflorescence, spikelet. So it is very important question and you must have known about this one. Spikelet is the special kind of the inflorescence which we find in case of the Poaceae family. This is the characteristic inflorescence in the Poaceae family. And here we see. So this is the structure of the spikelet here. In the spikelet, we see the structure pedicel and glooms. Okay, and these are also called as sterile glooms. Okay, the glooms are the structures without inflorescence. Okay, without flower, they don't form the flower. So these structures are called as outer glooms. These are the sterile structures, and the fertile structures. These are also called as bracts. Okay, glooms and lemma, palea, all these are also bracts. The sterile ones are called as glooms, and the fertile ones here you see outer gloom or outer bract is called as lemma. The inner bract is called as palea. These are the fertile structures, and they are present in they form the flower here. Okay, and lemma is it is larger than the palea. That you have to remember. Both are the structures, both are the bracts which are present, and the outer bract which is larger than the palea is called as lemma. So this is, and also you have to see about the on. On is a extension of the palea. Okay, and rachilla. So it is the axis of the inflorescence and each floret. So this is about the spikelet. You should remember that it is a characteristic inflorescence in case of the Poaceae family. And next, when Bateson and Punnett made a test cross between F1 individual of sweet pea having blue flowers and long pollen with plants having red flowers and round pollen. So they obtained 7:1:1:7 instead of 1:1:1:1 ratio. What does the results indicate? So in the genetics, you must have read about the test cross or double test cross. So 1:1:1:1 ratio, it indicates the double test cross. Usually in the Mendelian genetics. Mendelian genetics are different from the non-Mendelian genetics. These are the deviations. Non-Mendelian genetics are the deviation from the Mendelian genetics. Because as per the Mendel experiments, all the characters, the genes which are related to the 7 pairs of the characters, they belong to 7 different chromosomes. So there is no matter of linkage. There is no linkage because all the genes, they are related to different chromosomes. As per the experiments of the Mendel, what he studied, those genes or characters are related to 7 different chromosomes, 7 different pairs of the chromosome. So there is no point of discussion about the linkage. But later, several other scientists like Bateson and Punnett, they made some experiments, test cross. They got the results which deviated from the Mendelian experiments. So instead of 1:1:1:1, they got the 7:1:1:7 result. So why? What is the phenomenon related to this? That is the question. Linkage Independent assortment, crossing over, epistasis. So here independent assortment is related to Mendelian genetics, Mendel's experiment. So it is the wrong answer. And crossing over, epistasis, and linkage. The best answer here is most suitable one is linkage. So here if you see the linkage experiment, so what is linkage? First we see. Each chromosome contains more than one gene. Okay, each chromosome there are several genes, and the genes for different characters may be either situated in the same chromosome or in a different chromosome. Okay, when the genes are situated in different chromosomes, the characters they control appear in the next generation together or apart depending on the chance alone. They assert independently. Yeah, if the genes are located in separate chromosomes, then there is a independent assortment as per the Mendel laws. But if the genes are located or situated in the same chromosome, okay, but if the genes are situated in the same chromosome and are fairly close to each other, they tend to be inherited together, just like the people in the same boat. Wherever that boat goes, those persons which are in the same boat, they reach the same destination. Just like that, here, if the genes are present in the same chromosome, just like the attached, just like they are attached, they are inherited. Okay, together, they are inherited together. That phenomenon is called as linkage. Okay, this type of coexistence of two or more genes in the same chromosome is known as linkage. So, those this linkage phenomenon is explained here. Here you see in the chromosome 1A, 2A genes are there, and here 1B, 2B genes are here. These two are the homologous chromosomes, and here 1A and 2A genes are relatively little far away. Okay, so there is a chance of crossing over. When the crossing over occurs, the genes are away, the genes are separated, they are not close, then there are the chances that they can be separated by the genetic recombination. Here you see, during the crossing over, what happens? The homologous chromosome parts are exchanged between the non-sister chromatids. So that is called as crossing over. So here you see they are separated. 1A, 2A, 1A, 2A. Here in this structure we see 1B, 2A and 1A, 2B. Some new combinations we can observe here due to the crossing over. But in case of here, if they are linked, that means here you see the 3A, 4A are very closely arranged in the same chromosome, in the homologous chromosomes. So the chance of separation between the 3A, 4A or 3B, 4A is very less because they are very closely arranged. So this phenomenon is called as linkage. That is what observed here. Next question is, which of the following is a phytoplasma disease? Earlier I discussed already. Mycoplasma is given in your books, but phytoplasma, I think it is not given in your books, but that is the right word, phytoplasma. Phytoplasma also just like the mycoplasma, but it causes disease in the plants. And what is the disease caused by phytoplasma? Angular leaf spot of cotton, citrus canker, little leaf of brinjal, gummosis. Which is the right answer? Little leaf of brinjal. It is already given in your books. Little leaf of brinjal is caused by mycoplasma, but better than the mycoplasma, we have to choose phytoplasma because it grows in the, it is located in the phloem of the plants. It causes the diseases like the wilt diseases or little leaf of brinjal is caused by phytoplasma, and angular leaf spot of cotton that already we discussed. Okay, Xanthomonas malvacearum, Xanthomonas oxynopodicitri, It causes citrus canker. So like that, you have to remember the plant diseases and pathogen names. And now peptidoglycans are present in the cell walls of, already we discussed, they are present in the cell walls of the bacteria. Okay, and that is the right answer. And mycoplasma or phytoplasma, they don't have the cell wall, so there is no question of the peptidoglycan. Peptidoglycan is present in the cell walls of the bacteria. That is the right answer. Next question is rhodospirillum is a. What is rhodospirillum? We already discussed. Okay, purple sulfur bacteria and purple non-sulfur bacteria. So where uh, the we see the nutrition in bacteria, there are different kinds of the nutrition in bacteria. Photoautotrophic, okay, photoheterotropic, chemoautotropic, and chemoheterotropic. So these are the different modes of nutrition in bacteria. So in case of the rhodospirillum, which is a purple non-sulfur bacteria, I already discussed in the previous classes about the purple non-sulfur bacteria, because these purple non-sulfur bacteria, they don't rely on water. They don't take water. Instead of water, they take H2S. That's why they are called as this bacteria. But they release the sulfur instead of the oxygen. Okay. And about the rhodospirillum, it is a photosynthetic bacteria. That means it utilizes the light. It is also called as chemoheterotropic. Sorry, photoheterotropic bacteria. Photo means it utilizes, because in the photosynthesis there are two sources. One is energy source, another is carbon source. What is the energy source here? Light. This rhodospirillum, it has the photosynthetic pigment, bacteriochlorophyll. It has bacteriochlorophyll pigment. So it utilizes the light for the photosynthesis. The source of energy is light here. But the source of carbon is not carbon dioxide. If it is the carbon dioxide, it is called as photoautotrophic bacteria. But it is not the photoautotrophic. It is photoheterotrophic, because the source of carbon here is the organic matter. So this rhodospirillum utilizes light and organic matter as a source of carbon. Light as the source of energy, organic matter as a source of carbon. So it is called as photoheterotroph. So next one is, who crystallized TMV for the first time? TMV is the tobacco mosaic virus. Who crystallized it for the first time? You know, that is W.M. Stanley. He crystallized the TMV for the first time, and he told that all the viruses are the made up of protein crystals. He described them as the protein crystals. But later, Borden and Peary, actually Stanley got the Nobel Prize for the crystallization of the TMV virus. But later, Borden and Peary discovered that viruses are not only made up of the proteins, but also made up of the ribonucleic acid. Okay, so they are called as ribonuclear proteins. Okay, ribonuclear. Borden and Peary, they discovered that virus is made up of both proteins and nucleic acid. So they are called as ribonucleoproteins. And what he discovered? Twart discovered the bacteriophages. Okay, and Adolf Meyer, what he discovered? He discovered that these viral particles are contagious. That means they spread from one organism to another organism. So actually the question to the answer to this question, like the who crystallized TME for the first time, the answer is Stanley. But sometimes they may ask about the who discovered that virus particles contain both the proteins and nucleic acid. The right answer will be Borden and Peary. And if they ask about the who discovered bacteriophages, what? Who discovered that virus, they are contagious. That means they spread from one organism to another organism was discovered by Adolf Meyer. And next one is, which type of sexual reproduction is present in Zygomycotina? Zygomycotina, they belong to fungi. Okay. And what is the phenomenon we observe there? Gametangial copulation. So in case of the Rhizopus or Albugo, in our chapters, Albugo is given, but Zygomycotina means sometimes Rhizopus also they may give. Both belong to Zygomycotina. We observe the gametangial copulation. And next, who first recognized fungi as a separate kingdom? That is the Whittaker. Whittaker, he explained about the 5 kingdom classification. So he explained, he separated fungi as a separate kingdom. So the right answer is Whittaker.